Welcome back, friends. We are talking about mass spectroscopy, and we have discussed about the ionization process. Now we have discussed about the basics of mass spec, and we have discussed the process, step-by-step process of how mass spec is done, and then we have seen how to analyze the mass spec graph, which is generated by the computer system. Now, one thing we haven't discussed about yet, that is the different type of mass spec nowadays which are available, and in this video we'll be talking about two such type of mass spec instruments and analyzers, which are called, one of them is called MALDI and another is called ESI. So we'll be talking about MALDI and ESI. Though they are different things, they are utilized in different mechanisms in different reasons. We'll be seeing all of them. Now let us first talk about MALDI in this particular part. MALDI, the full name of MALDI is matrix assisted, matrix, M for matrix, A for assisted, laser desorption ionization spectroscopy, or sometimes another term TOF is attached to the MALDI. It is called the MALDI TOF. So we call it TOF, means time of flight. Okay. So matrix assisted laser desorption ionization time of flight. It is called the MALDI TOF. It's a mouthful of name, right? But the process is extremely simple, and you are about to know the process. And what is the process of MALDI TOF? I'm not going to discuss about again the whole mechanism of mass spec, but the simple region where every of different mass spec differs are the source region and the process of ionization. This part is varying from time to time. Otherwise, the main principle remains the same. That means we need to generate the parent ions, right? Now, when you generate the parent ions, the second step from all the different type of mass spec will be the same, because the parent ion will be moved to the magnetic field outside, and then it will behave differently in the magnetic field. By changing the magnetic field, we can select a particular type of fragment ion species from there, and as it is traveling the analyzer, those molecular ions will further be divided into fragments. We call them fragment ions. Then those fragment ions are going to hit the detector, and detector will detect it. Now in this case, the varying thing in any case of MALDI or ESI, now if I write here ESI, the full form of ESI is electro spray ionization. So it's called electrospray ionization, and sometimes GS is attached to it. It is called GC is attached to it. It's called gas chromatography. Okay, I hope you all know what chromatography is, so I'm not going to talk about that. So GC or HPLC is also attached to it. HPLC means high performance or high pressure liquid chromatography. Okay, so these are all about the abbreviations. Now let's look at the procedure. Now procedure, the major thing I have discussed, remains the same, but the difference is the generation of the molecular ion, because we require this, right? We require both of this process will require this. This is the molecular ion, and to generate this, all the things are set. So we are setting up all the things, all the instruments to get this part, because when we get this part, it will further fragmentalize into something like that. So we must have this parent ion first. These are the fragment ions. So we need to generate it, and we need to generate it from a sample or a molecule of our interest. Now this is our goal of generation of that. Now the process will vary from only the conversion of M to M plus, and here also the M to M plus. So in any kind of mass spec, this part will vary, rest of the part remains the same. Now we are going to see in case of MALDI top how the M is converted into M plus dash. Now how it is converted? In case of MALDI top, what we are doing, we are utilizing these molecules or sample of our interest. Now we take our sample and we place into a matrix. Now the matrix here is made up with some chemical. Now the chemical we utilize to produce the matrix here is generally 2,4-benzoic acid and cinnamic acid. So this benzoic acid and cinnamic acid, they act as the chemical cross-linkers to produce the matrix. Now we provide the sample onto the matrix. The matrix at the beginning is liquefied, then we press the sample, and then we allow this to be solidified, and it is solidified and produces a sample with matrix. Now the solid matrix is there and sample is entrapped onto the matrix here, which is made up with cinnamic acid and benzoic acid. Now this benzoic acid and cinnamic acid, they are having the aromatic rings. Now having these aromatic rings are very, very important. Having aromatic rings are very important for this process. Now what we have discussed till now, the samples are entrapped into a matrix. Now this is called the sample preparation phase. After that, what we need to do, we need to utilize laser to hit onto this sample. Okay. Now here one thing is very important, that the sample and the matrix and the components for matrix like benzoic acid and cinnamic acid and the ratio of taking them. Now here you must know that the ratio of taking sample and matrix are very, very different. Like the sample is to matrix is 1 is to 10,000. That much difference is there. 1 is to 10,000. So the matrix components are taken 10,000 times than the sample, because we are utilizing laser. If we utilize much more sample and less matrix, the laser is going to hamper our sample. We don't want that, because we need other matrix components and they contain the aromatic rings. So when you hit them with laser, they are going to get the energy from the laser, and as they are getting the laser energy, they will transfer this laser energy from them to the sample. But why we directly not hitting the sample with laser? Because it will destroy our sample, right? So we are hitting the sample to the matrix components, then the matrix components are going to get the energy from the laser, and they are going to provide the energy to the sample. And as they are providing the energy to the sample, so if I draw the matrix here, so say this is the matrix and here is our sample. Now they are entrapped, attached here. We are hitting them with laser. As we are hitting the matrix with laser, they are going to provide the energy to the sample, and as they provide the energy to the sample are getting kicked out. Okay, so sample particle are getting kicked out because they are charged now. So as they are charged, I am denoting it with charge, but this is not the exact. The charge usually is of positive charge, so they are getting charged, and as they are getting charged, they are dissolved from the matrix. That's why the term says desorption. They are dissolved from the matrix. They are just coming out from the matrix because now they are charged. They won't be entrapped into the matrix anymore because they are now getting excited, and then the sample are getting molecular ionized. So the samples then will generate this M plus molecular ion. So this is the procedure. We are not directly utilizing laser to the sample. We are instead hitting the laser onto the matrix. For that we need to prepare the matrix with some of the chemical compounds which are having aromatic rings. The compounds are benzoic acid, cinnamic acid, and then they will transfer the energy to the sample. Samples are getting ionized and they will dissolve from the matrix, and we will take them and we get the molecular ion. So again it is called the molecular ion. These are called fragment ions. Okay, now let us look at the ESI process. Now this is a simple process. Now we can utilize it with different analyzer. TOF is a time of flight. This is a type of analyzer. This is a type of analyzer and detector that we can use. And after the using of it, we can look for the graph that are giving us, or that is provided to us. Okay. Now here, in case of ESI or electrospray ionization, the process is different. Now here we also have the sample. Now here we take our sample. So here this is the sample. The sample is coated with a liquid matrix. Not as a matrix, this liquid solvent we can say. So here this blue colored region is a liquid solvent. Now this liquid solvent. So what we produce a micro droplet, and inside the micro droplet we are having our sample. We have produced a micro droplet. This is called a micro droplet. Now then, in case of this ESI, suppose this is the source. So whatever thing we have discussed had happened in the source region. Now here the sample is prepared as a micro droplet surrounding by the solvent. Now in this case the source region is having the high pressure content. So we are giving high pressure to the source region as well as heat to the source region. Now the high pressure provided in the source region will eventually going to heat this micro droplet. Now as they are hitting this micro droplet, then what is going on, this micro droplet and the solvents are getting evaporated, right? Because these are the solvents, they are getting evaporated. Now as they are getting evaporated, as this sample or as this total micro droplet is going to get into the analyzer, they are getting much more heat, they are getting evaporated, so a very little amount of solvent will left, and finally what we get our molecule, we get our sample, and the sample is getting ionized. Now if we utilize molecule directly without having any coat of the solvent in that high pressure, that is again going to hamper our sample. So we need to take the sample and coat it with a solvent liquid layer, and then we utilize into the pressure, so that if the pressure is giving the heat to this micro droplet, water will evaporate, not water, because this liquid solvent is also made up with water and nitrous water and also some chemical component, I just forgot the name. But as it is going, the liquid solvent is made with water and the chemical component in 1:1 ratio, and as it is traveling from the source region to the analyzer, the solvent part is getting evaporated. It will lead to the generation of only molecules, and as it getting heat, it will produce this molecular ion. So that's how it can generate molecular ion. Okay, so constant heating due to the pressurization is going on here. Now important fact about utilization of ESI is that we can utilize ESI in detecting large biological compounds such as peptides. So we can take peptide change, we can take DNA, so we can analyze peptides, DNA and all these things for the analysis utilizing this ESI technique. So that's why this is very much important. We don't need to fragmentalize these peptides, because in other type of like this MALDI processes, we must have fragmentalized that first. We must cut the peptides into smaller fragment of peptides or amino acid sequence, then we can analyze it with MALDI. But in case of ESI, we can actually go with the larger macromolecules. Okay, so this is giving us the advantage, and for this process also we need a chemical environment, and the chemical environment provided in this case is due to the N2 gas in the whole source region. But in case of MALDI, here we won't require any nitrogen gas. Instead we need to have a vacuum, a highly controlled vacuum to have this process. But in this case in case of ESI, we required N2 gas. Now after the whole process is done, as we are utilizing peptides and DNAs, which are large molecules, large biomolecules, macromolecules, we need to attach some of the other analyzers except for this mass spec, like the gas chromatography or the HPLC analyzer, a high performance or high pressure liquid chromatography analyzer, to finally detect the type of chemicals we are dealing with, because the molecules we are dealing with is, in this case, they are pretty large. Okay, so you can utilize this ESI process for these large compounds, we can utilize this MALDI-TOF process for the smaller compounds, but both the type of, you have seen that they are differing in their mechanism of the production of molecular ion from the sample, but rest of the process will be the same. After the production of the molecular ion, it will enter into the analyzer, it will further go and divide into the fragment ions, and then the fragment ions will behave into the magnetic field differently, and then we can select the type of fragment ions, and they are going to hit onto the detector, and they are going and we can detect the presence of few fragment ions, and by detecting the presence of fragment ions, we can actually tell what kind of chemical molecules we are dealing with. Okay, so this is about MALDI and ESI, the different type of mass spec, and I hope this will help you. Thank you.